I think if I press.
Yeah, it's recording. We're
All
good.
right, perfect. All right, so great group today. Great turnout, guys. Today we'll do protein misfolding. Um, here we go, John. Let's get started. I'm not about to slide. Oh,
Are you muted?
no, I'm not.
Are you sure?
Yes.
Okay. Okay. Little throwback to 285 here. So I've argued with Jen in a review session about this topic before. She was right. So I'm not proud. I'm not. I'm not proud. I mean, I'm not proud in a sense of like not admitting. Therefore, Jen was right, of course. Huh? Anyways, primary, secondary, tertiary, quaternary structure. Primary, it's but the sequence of amino acids. And they are linked together by this convenient thing called the peptide bond formation. What's a peptide bond formation, Neha?
Between the carboxyl and no,
Not the
wait.
carboxyl.
Oh,
Mhm.
I know what a peptide bond is, guys. Don't put me on the spot.
I already did.
It's just
What's the type of reaction?
it's a covalent bond, yeah.
But what's the type of reaction?
There's a name for it.
Yeah.
All water.
Oh, it's dehydration.
Yes, it's a yep. Let's drop our smoothies, guys. Yeah, so then peptide bond formation forms your sequence of amino acids together. Yay, let's go. Okay, secondary structure is when you start getting a little fancier with it. So you get these things called alpha and beta helices and also beta sheets. And this involves interactions by atoms in the backbone of the amino acid chain. So it is not R group interactions. It is only within the backbone. And every alpha helix takes about six amino acids for structure of the helix to continue. And if you have a proline, you're going to break that helical structure, as we talked about last week. And then beta sheets have configurations where R groups that are of a similar polarity will stick towards the same direction, and they'll protrude away. Oh, in both of these situations, they'll protrude away. Um, I started using it because people from use ChatGPT and it's actually like really good. Yeah. Um, but they will protrude away from the backbone. And so if it's a polar R group, they will tend to be facing the same direction as other polar R groups. Same goes for nonpolar. Tertiary structure. Okay, so this is now when we start getting to the three-dimensional shape of proteins. And so as we're going to talk about later a little bit, um, structure is essential for a protein's function. And so here we're ensuring that the proteins are folded on their specific three-dimensional conformation. And so you have R group interactions holding these proteins together, and you have polar non-covalent bonds holding this structure together, and you also have parts of the protein folding independently from each other. Those are your domains. And so you'll have characteristics of R groups that are nonpolar, hopefully probably staying within the middle of the protein so that they're not exposed to an aqueous environment. And the opposite goes for the polar amino acids. And so that is tertiary structure. And quaternary structure is when you need two or more peptide chains in order to form a protein. As an example of this, I have that beautiful picture of a antibody that has four different chains. They have two light and two heavy chains, and they're all in different colors for better exemplification. And then you also have that great looking hemoglobin at the bottom right. This projector kind of sucks, but the blue, um, there are four different chains in hemoglobin. Um, and so there's the blues and there's the reds. And there should be an iron atom in the middle there, but we're not gonna complain. Um, But yeah, so two or more peptide chains that form a protein together. Any questions, comments, concerns? No? Okay. So, I'm gonna talk a little bit more about the types of interactions that are being formed. So, common ones that you've probably heard, all of these. And so we just want to exemplify a couple of them a little bit more. So hydrogen bonds aids with the formation of helices and beta sheets, also beta helices that are less common and will be very prominent for protein structure and homeostasis as we delve deeper into the content of this week. Van der Waals interactions, which is a type of non-covalent interaction that happens between nonpolar groups. And they're basically just like the R chains are just gonna like move around because of Brownian motion, and they just happen to be near each other. So that's the interaction that they have. Backbone angle preference. So you have different factors that will affect the way that the angles for bonding will be held. And you can think about steric strain, and you can think about neighboring atoms that might interfere in such ways, and how nonpolar amino acids will repel away from polar amino acids and such things. And so for the most optimal angles to be held, you are hoping to have the most optimal three-dimensional space for that to exist. Hydrophobic interactions, a little bit of what we were talking about with van der Waals, but you're making sure that they're being tucked away into their hydrophobic pockets in these proteins. If you ever tried mixing oil with water, if you're cooking pasta and you have a layer of oil on top of the pan, this is because your nonpolar lipids cannot interact with the water molecules, and so they are sticking together because they're like, oh crap, I don't like that. If you're not using oil while cooking pasta, you suck at cooking. And chain entropy is just talking about any type of shift in the folding can result in a large loss of entropy for the specific molecules. And here we could talk about proteostasis, about how often a protein has to be upregulated, misregulated, so that it can continue on with its functionality. We could talk a little bit about loss of function and gain of function, but that will be further along in the presentation.
In that, in those water molecules,
Mhm.
the bond between, like, intramolecularly between the,
Oxygen, the hydrogen,
and then intermolecular,
it's a hydrogen bond.
why? Why isn't that hydrogen monophilic? Is that a camera question?
Well, it's because the water molecules are covalently linked, but when they're interacting with different molecules,
And the hydrogen
It's-
bond isn't a bond. It's technically just a dipole, right?
it's a dipole-dipole.
Yeah.
So it's a temporary dipole-dipole.
Basically.
Yeah, because the hydrogens have slight positive charge and the oxygens have slight negative charge, so then the.
Well, the oxygen have a very negative charge.
It's not a full negative charge, but
Mhm.
anyway,
It's like that sign that looks like a D.
but then you can also make technically hybrid bonds with like other things. Okay.
Yeah.
So I need a bond with itself, right?
Like within it.
Like, yeah. Oh, not with the- like protein. Yeah.
Well, if you have two hydrochloric acids, it's the same thing. Like the C and the L will form a covalent bond, but then the other H will bind with electrostatic interaction.
Yeah.
Did you ever change entropy?
No.
And
No. So let's say that there's steric hindrance from a larger, like let's say there's a phenylalanine that's not supposed to be there. It'll take more for like, let's say a chaperone protein that's aiding in the folding or like readjusting the folding of a specific protein for it to go in there and perform whatever magic it does in order to get the protein back. And so this use of energy, as the name implies, uses energy for you to get it back into correct conformation. But that's just what it's talking about.
okay. So that I remember that there's like lots of confirmations, and then the ideal one is a little, releases a couple of individuals.
Yeah, and I mean, if you have to take into account functionality, you have to take into account three-dimensional space, and also like how um like the I guess the net energy loss slash gained is in order to maintain like a conformation. Because like the most active form of a protein might not necessarily be the most energetically stable version of a protein. Well, because it might take more energy in order for like an enzyme to be active than a different conformation where it's inactive.
That's why it's
Changing conformations. Yeah.
regulated.
Sorry? Exactly. Anything? No?
Covered it all.
Okay.
Oh,
Go ahead, sir.
this might actually touch on what you're talking about. So the rate mechanism of protein folding, and so proteins don't have like an instruction book of how to fold. They kind of just seek the lowest energy conformation, and that can happen in like a bunch of different steps. And you can kind of see that in the graph over there. I mean, the projector sucks, but it'll start completely unfolded, and that's its highest energy state, and then it'll take random steps of folding and interactions and whatever to eventually reach that lowest energy state, and it's not going to go back up. Like that's naturally the order goes toward the least energy. And that's why you said it only releases like a couple kilojoules, because that's where it wants to be. So change to show more compact, yeah, have fewer conformations. So you have, because the lowest energy form is typically unique, you're going to end up with the lowest amount of conformational states at that point. So that's why proteins, will, if you have a specific amino acid chain, you can be in a bunch of different positions leading up to it. But once you get to the final folded state, it's a unique state. And that's why it'll, structure equals function. If your structure is always going to be that low energy state, your function is always going to be reliable, unless there's a mutation that disrupts it. And this is just called a funnel shaped landscape if it ever comes up in one of the papers. That just means like we're going to the lowest energy state. And yeah, so random steps, no instructions to reach it. And the proteins are generally found to fold in units of the secondary structures, so those beta sheets, helices. They're first developed, and then those interactions between them generally dictate how the protein's going to go into its lowest energy state. And this kind of helps it fold faster in a way. Like you build these basic units up first, and those helices, beta sheets, and then you just, those are larger sections of proteins, and you can fold it much faster. And yeah, like I said, multiple different routes can be taken to reach the same final protein structure. Some paths can be populated more than others. Generally doesn't mean it's like a right or wrong way. It's just, I guess whatever interactions are in the environment of that protein force, most of these molecules to go that way. Questions? Yeah.
Is it kind of I'm assuming proteins kind of fold as they're being translated.
Yeah.
So it's all.
Yeah, I mean, there isn't something that says, wait till we're all done translating so we can actually fold. Like as it's coming out of that ribosome, those helices are forming, the beta sheets are folding in, and once you have.
Because you can have proteins that have to go into like the ER or the Golgi, fold it and fold inside the ER or the Golgi.
Yeah.
There's proteins that you can go into have to be fully folded before they.
Yeah.
So kind of depends on localization.
Yeah. Good point.
It's the availability of chaperones.
Yeah, exactly.
Proteins filled with chaperones or some of them self-fold.
Somewhere.
Also, yeah, there's just so many different types of chaperones. Most of them have something to do.
Chaperones fold themselves.
I think we talked about this once. It's like, no, there's a chaperone for the chaperone,
Chicken would be
but
Well, but if every cell comes from a cell, then you don't really have that shit.
because you get some of the materials with it.
Like when the like in development, when the thing forms, it's not that like you're forming a thin air, like the sperm and the eggs are out like
chaperones.
and
Exactly.
like when the different germs form here, it's itself.
Yeah.
That makes a lot of sense.
It doesn't start quickly nothing. It's like, finish 2 cells first.
Yeah. Anything else? Sweet. So there's this because there's so many routes to, reaching the correct state, there's many opportunities for misfolding. And especially in disease-related mutations, the correct interactions, usually aren't ever attained. The cell is able to kind of combat this using, expressing chaperones that can guide the correct folding. And, be in response to the accumulation of proteins in general. And they can also force the misfolded proteins into controlled degradation and recycling. I mean, one more thing that I did not put up here is that, the cell, if it can't overcome these things, it just generally can push the cell into apoptosis, and kill it whole. It's better to have a dead cell than an uncontrolled cell
This is happening a lot. It's like ubiquitination happens a lot.
all the time.
And
Yeah.
it just goes into the lysosome and then you get free amino acids again.
Yeah, yeah. Yeah. And then I think John wants to talk about UPR.
Okay. Yes.
So, unfolded protein response, as we were talking about a little while ago, a protein's functionality is highly linked to and dependent on its optimal function or optimal structure. And so let's say in a situation where like the pH increased or like the temperature increased all of a sudden and the cell starts getting a lot of like these misfolds slash unfolds, it's gonna have specific responses to that, as the functionality of like specific types of proteins are more important than some others. And so the cell is going to focus on trying to like respond to the more, oh crap, I can't have this go wrong proteins rather than like some very, or not very less optimal, but like less important proteins. It goes in a matter of like, do I need to do this first or do I need to not do this first? And so you have specified genes that are developed specifically for this. So you have IRE1, you have ATF6, you have a couple other genes that the paper cites. And so in order for you to have the cell not be flooded in these different express proteins that are misfolding slash unfolding, translation stops and you're going to have a specific process for degradation starting in the ER. And so the associated degradation process or protocol starts, and this is mediated in the proteasome via ubiquitination. And if homeostasis cannot be reached, if proteostasis for the different proteins also cannot be reached, then programmed cell death is going to happen because, as Aaron was talking about, it's easier for you to have something go bad if the cell does not induce itself into apoptosis, and so it just chooses the better option of going into cell death. And it's not like this instant thing that happens, like I'm not gonna just like shut off. Like there are a bunch of like different mechanisms that need to be activated. It starts with the mitochondria. But it's a very controlled process so that like no damage is passed along to like progenitor cells. Good.
Yeah, that works.
Okay.
What else do we have? Oh yeah, so we have the heat shock response, which is kind of specific to the cytosol and the nucleus. So this is also a stress response like the UPR. This can be triggered by elevated temperatures, hence the name heat shock, or any other stressors that cause misfolding. So this could be like pH changes, mutations, anything of that sort. It's not restricted to heat. And there's a number of heat shock response proteins that are transcribed. There's no way you're going to know all of them. A couple important ones maybe that might come up in your reading is HSF1. This controls the expression of other HSR protein synthesis. So it's kind of like the master one that kind of dictates what else is synthesized downstream. And it's usually bound to other proteins in its inactive form. So that's important because it kind of tells you that it's always there, like it's always expressed, except the cell keeps it in check by keeping it inactive. So when it does need it active, it immediately can just, induce it to unbind from those other proteins and, start function. It doesn't have to like transcribe it fresh every time there's a response to a stress. Yeah, and then it goes on to bind different heat shock elements along the DNA, induces transcription, so we can get those downstream components. And then the HSP70 is what, it's not the only one, but it is one that mainly plays in the facilitation of the refolding.
HSP stands for heat shock protein.
Yeah, sorry.
Found out.
But it's not only shock, it's other environmental changes that you trigger.
I mean, yeah, heat shock is like the name for it, but like, pH changes, mutations, those all can. It's just basically a stress response that also includes heat. Heat denatures things, ruins things.
They also like catalyze reactions with like activation of enzymes. So there's like a fine line between your enzymes working better because of like elevated temperatures and them not being able to exist.
Yeah,
Which is kind
it is. We cool? All right. So this is kind of getting more into what the paper discussed. How does protein misfolding arise and lead to disease? Well, we're gonna go through all of these, but this was the nice little figure in that paper. I thought this was great, by the way. It just basically gave the whole paper in one image. So very cool. We kind of already talked about energy changes, so we'll go to the other ones, but improper degradation. So degrading proteins is essential to preventing any sort of accumulation. So if you have a misfold of proteins, you want to, either fix it or promote degradation. So it's not a problem anymore. But if you have an overactivity of this degradation, so you're degrading proteins without needing to, it also causes some problems. And this is kind of what we see in cystic fibrosis and what the paper kind of gave as an example. So this is where a cystic fibrosis transmembrane conductance regulator, CFTR, is so this is a plasma membrane chloride chain, and a mutation in this where you lose phenylalanine residue changes the wild type function because you need this residue to actually the lack of this residue is what causes the protein to misfold, and because you have this misfolded CFTR, which I think still functions fine, but like the fact that like the body is just degrading it unnecessarily causes problems. I can be fact checked on that. So basically you just don't have the CFTR anymore because the body's just constantly degrading it. And with that degradation, you go from healthy lungs to lungs with thickened mucus because that chloride channel is not working right. I think I said chain. I meant to say channel. And typically treatment for this involves disrupting the activity of degradation. So chaperones AHA1 and CHIP. Those are some chaperones that are involved in the degradation. If you disrupt the activity of those, you're not going to get the degraded CFTR. And because, like, I think this is why I thought the mutated CFTR still works fine, is because, you degrade these, then you see improvement because those chloride channels still work.
Is it what's it called? Nonsense? No, not nonsense, the one is it just the one phenylalanine that's deleted, so everything downstream is just wrong? What is this?
It's a frame shift.
Yeah.
I think it's a missense.
I think so too.
Is the phenylalanine amino acid deleted, or what is?
I think it gets replaced.
Yeah.
I think it's a missense.
When papers say like residue, does
That just means one amino acid.
I would assume a frame shift messes a lot more things up, so you wouldn't get a
If you
functional
have,
protein.
like your solid or like your solid.
Yeah, so I
should have born,
Yeah, that's a frame shift. That's
Yeah.
a frame shift.
no, I think this is just replaced because you maintain the other reading, it's just that one has changed.
I think it's a missense.
Pretty cool.
is this? Is it fair to say that this is like gain of function or gain of toxic?
Oh,
Oh, it's a deletion.
it is?
Frame shift.
Oh, wow. Alright. And yeah, it is a friendship, but, your question? I don't,
Or is it not fair to even say it's a gain of function or a loss of function? It's just like
Um,
selective. And the reason it is the treatment because it's kind of still a little bit functional, and so you don't need to delete all of it.
yeah, like you need this, or else you end up with that,
Even with
the
the.
mutated.
treatment,
Yes,
because
I'm pretty
it's
sure,
Still somewhat functional.
yeah, I'm pretty sure that's the case. I'll fact check myself after this. Yeah, and that's why like treating the chaperones helps. Because you just, you block degradation and you leave that mutated protein still there, it'll work. Um, now, as far as your question, do you think this is gain of or loss?
I think it's loss function of gain and toxic. Because you're overly degraded. You lose the function of. the
Yeah.
mutation.
But it's still mutation. That's
But
all.
like, but you're, because
It's
you're
just all
overly
like-
degraded.
Yeah.
You're not.
So it would be loss of function, not gain. It's the same.
It'd be loss function and gain toxic.
It can be both. Yes,
Okay.
because you're considering like the protein and then downstream, like also like gain of toxic function because,
Got it.
okay, cool. All right, next. So we also have improper localizations. So proteins have to localize the specific parts of the body to complete their function. And the localization, and it's just kind of like 285 we talked about, it relies on the correct folding because those, the localization sequences are embedded in the protein. It needs to correctly fold so that the chaperones can recognize this and bring it where it needs to go. So this is one of the cases, not the only one, obviously, but one of that incorrect folding can lead to a loss of function in the protein and a gain of toxic function. GOTF. An example of this is the alpha-1 antitrypsin. This is a protease inhibitor that functions in the lung as well, kind of similar to cystic fibrosis. Except when if it's produced in the liver, when it does not have correct localization because of the misfolding, you're not able to get it out of the liver. It kind of just builds up in the ER of the hepatocytes. And because of that buildup, you're both getting a lack of the function in the lungs where it needs to be. So this is like causes damage in the connective tissues, but also that buildup damages the hepatocytes. They're not able to, it accumulates, they start, stress response can't fix and they start dying. So that's where that gain of toxic function comes from, but the loss of function also comes in the fact that it's not in the lungs.
There are some like mitochondrial diseases that happen because of this.
Mm.
I think there's only like 18 or something proteins that the mitochondria makes, but the body makes all the other proteins that are necessary for the mitochondria to use in cell respiration. And so if you think about like if there's a specific protein that's completely necessary for cellular respiration to happen and it can't make it into the mitochondria, then by default you have a toxic gain function of like a different protein that will not be fulfilling the exact necessity for that protein, and you also lose the functionality of irrigation.
Yeah. And so treatment for this kind of involves enzyme replacement therapy. So I don't know if this totally combats the gain of toxic function because it doesn't really make sense in my head, but definitely affects. Hey, perfect. It definitely affects the loss of function in the lungs because you just replace the protein with something that is actually able to be there in function.
And the enzyme we're replacing is this antitrypsin.
Yes, a functional localized version of it, correctly localized.
Is it the same trypsin that people use for extraction?
I have no idea. John?
Wait, what?
Is the trypsin the same trypsin that they use for RNA extraction?
Yeah. Anything else?
Is the reason that it's unable to be secreted from the ER because it has this folding?
Yeah, and so the misfolding kind of prevents the localization sequence in it to be recognized and, so it's not able to be transported by the chaperones. It kind of just stuck there.
And so when enzyme replacement therapy, does that get rid of the non-functional one as well?
Yes, yeah,
Because that like, I can see that the loss of function would be fixed with it, but
yeah.
what about the toxicity?
Yeah, that I don't know. I imagine you know, the buildup in the hepatocytes is still a problem, and there might be like all like compound therapy, so like this and that might work, but I just know that this treats the loss of function. I don't know about the gain of toxic function though. It would make sense that if those mutated ones are still there and causing damage though. So I don't see how this would cause fix growth problems. Yeah. Good. Okay. And then, ooh, dominant negative mutations. Okay. So this occurs when a mutant protein antagonizes the function of the wild type protein. And I actually don't know if anyone knows, like, why is this different from the prions that we kind of discussed in class? Does anyone know from the reading? Or wants to take a guess based on the definition?
I mean, are they specifically for brain? It's like they're general.
This is more like in a general functionality.
General functionality, yeah. So, I guess prions, they work by being, you know, this weird mutated form like that doesn't fold right of a protein, and specifically it will go to other healthy right folding, you know, correct folding proteins and make them misfold. This is different in that you have this misfolding protein, yeah, but it's not going to cause those healthy ones to misfold. It's just going to stop them from completing their action. So they're still healthy proteins, you know, the other ones that are not affected, but it's preventing them from completing a correct function, right?
The prions are contagious. But
Yes.
inside
Yes,
Yes.
yes,
Totally.
very.
Dominic, this one isn't. It's just like overpowering.
Yeah, I don't know if it's contagious or not. I bet there might be some cases where it is contagious, but like prions are the big contagious one. Prions are scary
Well,
because
prions, it's not that prions are the big one, it's that prions were the first one that were discovered specifically in neurodegenerative stuff.
and we still don't
I'm
have it.
gonna get onto like the hallmarks paper. Like it's scary because like most NDDs have a prion-like way of transmission. It's not that it's specifically just for prions.
Yeah.
That prions were the first one.
So dominant negative mutation, they can assume prion like.
No,
Yeah.
I don't think they have like organization and seeding. I think it's just they just get like the functionality is wrong.
Yeah.
And then they prevent it.
Yeah, they prevent. Um,
What were you gonna say?
I know that last year a paper came out. It's like the theory about the tau tangles and the tau little the seeds causal to make neurodegenerative diseases, prion-like things is wrong. Like that came out and was published. And then people, and they were like, that's probably why none of these treatments are working. Is that?
Well, but like it would be if it was strictly because of tau fibrillary tingles, sure we can make an assumption for it, but it happens with alpha-synuclein. It happens with like
It's
a bunch of other proteins. It's not just tau.
Yeah.
It happens with prions.
Yeah.
It happens with PRP. Like wild type PRP is regular, but if you have a misfolded PRP, and it touches the regular PRP, then it becomes prion configuration.
Awesome.
Yeah.
That's what I was
Yeah,
wondering.
yeah, yeah.
How many diseases?
Yeah, I mean,
it's
it's like
expressed.
most of them have like some sort of like protein oligomerization, misfold oligomerization, breakdown and seeded.
Yeah.
And it's like you'll have a concentration of these proteins, they'll break off and they'll just go to like they'll force themselves to be explored out. They'll they'll move on to different parts of the cell. It's like insane.
yeah. So quick example, this is P53. P53 is constantly expressed because it's a very important cell cycle arrest facilitating protein, DNA damage response. Definitely heard of it, I hope, right? Okay, cool. It's degraded by the proteasome in times of no stress, and this relies on the MDM2 complex, which is not that important, but it is there. In times of stress, P53 is stabilized and goes on to complete its function by tetramerizing. So it comes in, pairs of fours. Those come together, and those complete downstream effects of, cell cycle arrest, whatever, DNA fixing. But a mutation in the core domain of this P53 leads you to have this misfolded P53, and it can't tetramerize with other wild type P53s. and this prevents them from completing their function. So this is where you have like the case of, let's say like two mutated ones and two normal ones. Those two mutated ones, two normal ones will come together and tetramerize, but they're not going to tetramerize in the way that like wild type would, and so they're not able to complete their function. You know,
But just not in the right.
yeah, I think it's just they like bind each other and then like I don't know if do they bind each other? And then they like prevent each other, or they just like
I think they bind each other, but not in a way that complements each other's functions, so then the ultimate function. Because like the way P50, like I'm not like 100% sure about how the mechanism
Yeah.
for like DNA checking is done by P53. But I assume they have a way of sensing if there's like a lesion in DNA.
yeah.
Like if like the curvature of the DNA is wrong, or if there's an extra nucleotide, or if there's like a mismatch nucleotide, and they're able to like sense that, assuming that the way that the DNA would be read by the molecules would not be functional.
Yeah, and then the P53, the mutated one, kind of also evades degradation, very resilient. So while the healthy ones are being degraded and under no stress, this guy's still there. And a way to treat this actually was you kind of just flood it with more wild type P53. And when you do that, you're kind of decreasing the probability that these ones will have any effect. You know, if you just triple the number of normal P53, let's say, then your chance of having 4 healthy ones tetramerize increases much more.
That was kind of like an separate therapy.
Yeah, in a way, like those bad ones are still there, but you're just increasing probability that you're going to get the healthy tetramerization, and that will actually work. Good. All right. Okay, gain of toxic function. So this is the one where the conformational change in the proteins can lead to toxicity in certain parts of the body. We kind of saw 8 cases in the was it cystic? No, the other one. Improper localization. Yeah, that one.
Prion disease too.
Prion disease too. Yeah. The example I pulled was the ApoE. Do you call it ApoE or APOE? Apo.
I know, because I remember using.
Oh. Oh, really?
Yeah.
That's cool.
Well, if you're heterozygous, you're increased by like a little bit.
Yeah.
Homozygous, it's increased by like.
Like a lot. Okay,
It's also not the only factor. Not everyone with mutation gets it.
Right.
Not everyone
there's a lot. Yeah.
has the mutation.
Right. Okay, so the APOE is involved with the lipid metabolism. I know, but I picked the way I want to. No.
Okay.
How is it? APOE? ApoE. 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 Yeah. Yeah, it doesn't. John's just being big. ApoE is involved with lipid metabolism and transporting cholesterol transport, for example. And it comes in many forms encoded by different alleles. So this specific variant, the 4-number variant, is associated with disease and involves a change in the protein structure that promotes the formation of a salt bridge. This salt bridge is associated with altered lipid binding and an increased accumulation of A beta. This protein, the protein that's known for aggregating in Alzheimer's.
Neho, what's a salt bridge?
Don't.
Hmm?
Disulfide bond,
And what's that bond to?
sulfur and sulfur.
No, but why? Like, what type of bond is that making? Why is it so special?
It's pretty strong.
It's the strongest non-covalent.
It's a
reaction.
covalent bond.
Oh,
It's cool.
strongest covalent.
It's like
It's a regular covalent, but it's or covalent bond.
cysteines.
Salt bridge is not very strong.
No, it's a covalent. So
It's like the only covalent bond you'll get outside of the backbone.
Salts, because people refer to salts as like that.
such a salt.
Ionic bonds are pretty weak inside cells.
Because they're very reactive.
Yes.
And bad sometimes.
Yeah. I couldn't find a treatment for this. I think you kind of just deal with it. It's a bad way to say it, but like there's no really effective like preventative or treatment or anything. I guess You know, like you said, there's a lot of other factors that go into it. So you could, take a look at those, which I don't know, but no like treatment. But
I think they're trying a lot of gene therapy, they're trying a lot of.
Yeah.
This, that, but. Because it's so multifaceted, no one really knows, but there's a lot of money.
yes,
Guaranteed job. you
Agreed.
Get funding.
Agreed.
It's stunning, funny.
all right, cool. Any questions? Sweet. Okay, and last one. Amyloid.
Amyloid.
Amyloid accumulation. So amyloid fibers are these insoluble fibrous protein aggregates with a high level of cross beta sheets, because those cross beta sheets, pretty strong when they come all together and fold, and they can stack really nice, which makes that fibrous form. With enough of this accumulation, this can lead to the aggregation of entire proteins. So not only do you have these fibers now that are aggregating, but you have entire proteins that are, aggregating when together. And this is kind of like our big topic, and you'll see in the Hallmarks paper, this is a lot of, like, this will be discussed very heavily, but a lot of different diseases are attributed to these aggregation of proteins that stem from the amyloid fibers aggregating together. So Parkinson's, we already talked about Alzheimer's, type 2 diabetes, even cataracts in the eye, which are cool. I didn't even know, like the crystalline will, they will form aggregations, and then those proteins are aggregate, and that's why, you get this sheet that you can't see out of. Yeah. Yeah, prions. And then to combat this, antibody therapy has been shown to have somewhat of an effective course. And it does this by basically just binding those fibrous amyloids and preventing them from, like, if you have something wedged in there, it's not gonna stack right, it's not gonna aggregate. Now, as far as, how effective this is, I have no idea. I doubt that effective, because we still see, like, a lot of research in this and in all these diseases, and, no fully effective course of treatment. And I'll take questions, because I think, yeah, that's our last one. Sweet. So I will stop the recording somehow. Ah. There.